Organ-on-a-chip breakthroughs represent a revolutionary convergence of biology, engineering, and microfluidics. At their core, these devices are tiny engineered platforms that mimic the functions of human organs by recreating the physical and biochemical environments found in our bodies. Imagine a miniature lung, heart, or liver fabricated on a small chip where tiny channels circulate fluids, just as blood flows through our veins and capillaries. This biomimetic approach offers researchers a unique window into human physiology without the complexities of whole organism studies. One striking example is the lung on a chip. In this device, scientists designed two parallel microchannels separated by a flexible, porous membrane. One channel simulates the air-filled space of the lung's alveoli, while the other mimics the blood vessels. By applying cyclic mechanical stretching to the membrane, researchers replicate the rhythmic motion of breathing. This innovative setup allows for the detailed study of respiratory diseases, environmental toxin effects, and even the response to viral infections in a controlled setting. Could this be the key to unlocking more effective treatments for conditions like asthma or chronic obstructive pulmonary disease? Many experts believe it just might. Similarly, heart-on-a-chip platforms use microfluidic channels to recreate the dynamic environment of cardiac tissue. By integrating human heart cells into these devices, scientists can observe how these cells beat in synchrony, respond to stress, or react to new drugs. This is particularly valuable for screening compounds for cardiotoxicity, a leading cause of drug failure, before moving to clinical trials. Imagine the implications: faster, safer drug development with a much lower risk of unexpected heart side effects. In the realm of liver-on-a-chip technology, the goal is to emulate the liver's crucial role in metabolizing drugs and toxins. These chips use networks of tiny channels to simulate the liver's blood flow and cell interactions, enabling precise analysis of how substances are processed in the body. This not only aids in understanding liver diseases, but also provides a more reliable model for testing the safety and efficacy of new medication. The potential here is enormous. By predicting how a drug will behave in the human body more accurately, researchers can streamline the path from the laboratory to the pharmacy. Collectively, these organ-on-a-chip systems are transforming disease modeling and drug testing by offering a more human-relevant alternative to traditional cell cultures and animal models. They bridge the gap between in vitro experiments and clinical reality, leading to insights that might otherwise remain hidden. As we continue to refine these technologies, intriguing questions arise. Can we eventually link multiple organ chips to simulate entire bodies? How will these advances change the way we approach personalized medicine and complex disease treatment? Each breakthrough not only enhances our scientific understanding, but also pushes the boundaries of what's possible in medical research and patient care. Organ-on-a-chip devices are microengineered systems that replicate the functions and structures of human organs on a small chip. They integrate living cells into a microfluidic environment that simulates blood flow and mechanical forces found in real organs, aiming to bridge the gap between traditional cell cultures and human physiology. Researchers create a lung on a chip where human lung cells are cultured in channels that mimic the stretching and contracting motions of breathing. And imagine building a miniature working model of a car engine on a tabletop where every moving part functions just as it would in a full-size engine. Microfluidics involves the precise control and manipulation of tiny volumes of fluids in channels that are often only a few micrometers wide, enabling researchers to recreate the dynamic environment of human tissues, including the flow of blood or other fluids, and allowing rapid, controlled experiments that mirror physiological conditions. A heart on a chip uses microfluidic channels to simulate the pulsatile flow of blood, allowing the study of cardiac cell behavior under realistic conditions. And think of microfluidics as a network of miniature plumbing pipes that deliver just the right amount of water to each plant in a carefully designed garden. Organ-on-a-chip platforms are designed to closely mimic the physical and biochemical environment of human organs, by recreating key features such as tissue interfaces, mechanical motions, and chemical gradients. which is crucial for understanding how organs function and respond to various stimuli, including drugs and disease. A liver on a chip is engineered to simulate liver metabolism by incorporating multiple cell types and flow conditions that mirror the liver's natural environment. And imagine setting up a detailed model train set that not only replicates the tracks, but also includes realistic scenery and weather conditions, capturing the full experience of a real train journey. Organ-on-a-chip technology revolutionizes disease modeling by providing a more accurate model of human disease compared to traditional cell cultures or animal models, as these devices allow for the recreation of complex disease states, such as inflammation or cancer, in a controlled environment. Researchers have developed a gut-on-a-chip to study inflammatory bowel disease, where the interactions between bacteria, immune cells, and gut tissue can be observed in real time. And think of disease modeling on a chip as having a virtual weather system that accurately predicts storms by simulating atmospheric conditions rather than relying on outdated models. Organ-on-a-chip platforms are transforming drug testing by providing a more realistic prediction of human responses to pharmaceuticals, enabling the assessment of drug efficacy and toxicity on human tissues before clinical trials, reducing reliance on animal models and improving both the speed and accuracy of drug development. A kidney on a chip can be used to test how new drugs affect kidney function, helping to identify potential toxicity issues early in the development process. And imagine testing a new recipe in a high-tech kitchen that perfectly replicates real-world cooking conditions, ensuring the dish will turn out well in any home kitchen. Organ-on-a-chip systems offer the potential for personalized medicine by using patient-specific cells to create customized models of organ function, which allows for testing drugs on an individual's own tissues, leading to more tailored and effective treatments and a significant step forward in predicting patient responses and optimizing therapy regimens. A tumor on a chip can be constructed using cancer cells from a specific patient, enabling oncologists to determine the most effective treatment strategy for that individual. And think of it like having a custom-tailored suit. By using your specific measurements, the suit fits perfectly, just as personalized medicine ensures treatments are precisely suited to your body. Recent breakthroughs have led to the development of multi-organ on-a-chip systems that interconnect different organ models to simulate whole-body interactions, providing insights into how drugs and diseases affect multiple organs simultaneously and offering a holistic approach essential for understanding systemic responses in a complex organism. Scientists have connected liver, heart, and lung chips to study how a drug is metabolized by the liver, circulates through the bloodstream, and impacts cardiac function. And imagine a miniature city model where every building, road, and utility system is interconnected, allowing you to study how changes in one area affect the entire city. Organ-on-a-chip technology overcomes many limitations of traditional cell cultures and animal models, which often fail to accurately mimic human physiology, by providing dynamic, real-time models that closely resemble human organ systems and enabling more predictive and reliable research outcomes. A brain on a chip allows neuroscientists to study complex neural networks and responses to drugs in a way that isolated cell cultures cannot replicate. And think of it like upgrading from a static, two-dimensional map to an interactive, three-dimensional simulation that offers a much richer and more accurate view of the terrain. The continued evolution of organ-on-a-chip technology promises to revolutionize healthcare by enabling more precise disease modeling and efficient drug testing, with future advancements potentially including fully integrated body-on-a-chip systems and further personalization of treatment strategies that will reduce healthcare costs, improve patient outcomes, and accelerate medical research. Innovative research is already underway to combine organ-on-a-chip devices with artificial intelligence, aiming to predict patient responses to complex treatment regimens more accurately. And imagine a futuristic control center that monitors and manages an entire ecosystem in real time, allowing for swift adjustments and optimal functioning. That is the promise of the next generation of organ-on-a-chip technologies.